So we have talked about two types of T cells, cytotoxic T cells and helper T cells. Let's look at these two types of cells separately. First of all, we'll talk about the cytotoxic T cells. Cytotoxic T cells provide protection against intracellular pathogens, for example, virus and also some bacteria that can actually enter the cell, host cell, and start multiplying there. So we have talked about that the fragments of these foreign entities, these pathogens, can be grabbed by the MHC1 molecule, which is present on the surface of all the cells, all the nucleated cells, excluding RBCs, of course, and it is displayed on their surface. These peptide fragments are read by the TCR, determining whether they are self or foreign in origin. How they do that, we'll talk about that later. So this is basically the function of cytotoxic T cells to go around and see if our normal body cells have normal proteins which they should have, or whether they have been infected by an intracellular host, or even in some cases, cancer, for example, results in expression of unnatural genes, gene products. They can also recognize that, and cytotoxic T cells can also destroy those cells. Cytotoxic T cells, as I've said, recognize cells that have been infected. These cytotoxic T cell receptor interacts with the class 1 MHC protein displayed on the surface of antigen presenting cell. Once T cell receptor encounters such a cell that is displaying a foreign antigen and that T cell receptor recognizes it, the T cell basically receives a signal which activates that particular T cell. The T cell, once it is recognized, it will proliferate and form a clone of cells. So it is going to, all the cells that are going to come from that particular T cell will have the same TCR specificity. So these T cells, once they are activated, they'll proliferate and make many, many copies of themselves. Once that happens, they will go around in the body looking for other cells that have been infected. During this process of T cell multiplication, T cells also become mature. And now they have become the effector T cells, cytotoxic T cells. They have gained the ability to destroy the cells which have been infected. We will see how T cells destroy the infected cells later. But here we basically wanted to talk about the T cell activation. Here in this photograph, you can see two T cells depicted in orange. I will point them out. Here are the two cytotoxic T cells, orange colored. The purple or pinkish colored cell is basically a cell that has been infected. This cytotoxic T cell interacted with the cell that was infected and resulted in its destruction. These small vesicles you're seeing basically are a result of apoptosis. That cell which was infected was instructed by T cells to go through apoptosis or destroy itself. We will see how that happens later on. But this cell has been completely destroyed, where this T cell is in process of destroying this pink cell, this larger pink cell. We can tell that by these blebs which are forming. These vesicles or blebs which are forming on the surface of this cell, which is a hallmark for apoptosis. We will talk about apoptosis later on. Here are a couple of electron micrographs on your screen. You can see the cytotoxic T cell interacting. Here's a cytotoxic effector T cell interacting with the host cell. It has bound that cell. The cytotoxic T cell has bound a cell which is carrying for intracellular pathogen, for example, a virus. Here you can see the cytotoxic T cell has already destroyed that cell. This micrograph is very interesting. Here you can see the smaller cell. This is the cytotoxic T cell attacking a cell which has been infected by a particular intracellular pathogen. Please notice this region. This is basically the centrum here of the cytotoxic T cells. As we have already talked about, centromere is basically microtubule organizing center. So the microtubule organizing center, as you can see, has shifted and is now adjacent to the infected cell or the target cell. The reason, the vesicles will now start moving along the microtubules, and these vesicles will be bombarded onto the target cell, and the target cell will be destroyed. I'll show you better photographs of that subsequently in more detail, that what is happening at the cellular level or the molecular level. Here's the schematic which depicts how cytotoxic T cells kill the target cell. Two strategies. First of all, the cytotoxic T cell binds the target cell which is infected. It releases contents which are present in its vacuoles. These vacuole contains proteins that can assemble in the plasma membrane of the target cell, and when they assemble, they form little holes. So cytotoxic T cell releases proteins that are making holes in the plasma membrane of the target cell, which are shown here in green, for example. Other proteins now can enter through these pores, and when they enter the pores, they activate other proteins inside the cell. These proteins are enzymes. Once they're activated, they can basically destroy all the macromolecules inside that particular cell, resulting in apoptosis of that target cell. So this cell has been destroyed. The second strategy of T cells is they can use Fas ligand. It is a protein which is expressed on the surface of the cytotoxic T cells. When Fas ligand binds the Fas, which is a receptor, you can think of it as a receptor, these receptors come together. When these receptors come together, the cytoplasmic domains of these Fas molecules result in recruitment of special type of proteins called capsases. Capsases are mildly or weakly proteolytic. A whole bunch of these capsases are assembled. They're near each other. Their weak proteolytic ability causes cleavages and results in active form of this capsase. Capsases are proteins that can also basically destroy different macromolecules within the cell. So here the T cell, cytotoxic T cell, has also destroyed this target cell using a different strategy. Here the strategy was bombarding the cell, the target cell, with vesicles containing molecules that will destroy it. Here it is done through signaling molecules, both achieving the same result. Here the question is, why doesn't these vesicles, when these proteins are released in the extracellular space, why don't they also destroy the cytotoxic T cells? That is something I would like you to look at, find it on the internet or use other sources and come up with that answer yourself. Also, effector cytotoxic T cells can release different molecules, signaling molecules. Interferon gamma, for example, once it is released, it causes the cells which are present in the around the infected cells to adopt a strategy that will prevent them or block the virus or the intracellular pathogen from infecting them. For example, these interferon molecules, gamma interferon molecules, they can allow cells to increase the MHC protein production. They also increase the proteasome subunits, which have ability to degrade different molecules, and also peptide transporters. These interferon gamma molecules, when they bind the receptor, they result in gene activation, and also which results in viral replication blockage using different strategies. So we have seen how cytotoxic T cells function and how they have evolved two different strategies to kill their target cell.